First of all, let me thank the organizers for the invitation. And after yesterday afternoon quiz and some conversation with colleagues, I decided to change completely my presentation. And so I am not sure how will be the presentation this time, but I think that will allow some kind of interaction with the audience. Well, so the papillomavirus family is very large. There are over 200 types, and the one involved with human disease are mainly the alpha papillomavirus, the beta and the gamma. This classification is based on using specific primers focused on the late one region. And with this approach, we can identify the different phylogenetic genotypes associated. But if we use the type of primers, for instance, using shorter primes, much more specific, then you can see an increase of number of types that we can identify. So actually, the number of papillomavirus genotypes is growing, especially for the gamma and for the beta type. Our conversation today is mainly focused on the alpha, because some of them actually have been associated with human cancers. Among the almost 30 genotypes which are involved in the alpha family species, then we have the first 12, which are mainly oncogenic, and specifically the HPV-16 and HPV-18. And actually, just HPV-16 is the most potent and is involved not only for the cervical cancer, but also with the other cancers associated with the papillomavirus, which we will see in a few minutes. For the other one, for the 68, 26, and so on, there are limited evidence or not very limited evidence in humans, but with a sufficient level of certainty. So these are classified as group 2A, 2B, and then as a 3, which means there are not enough evidence is for genotypes 6 and 11. Most of the oncogenic activity has been related to the activity of the two oncoproteins E6, E7, which actually play a major role in inhibiting two oncosuppressors, the P53 and the PRB, the retinoblastoma oncosuppressor gene. Now, the ones that are mainly associated with the cancer are part of the two species, A7 and alpha 9, and within the alpha 9, we will see, besides the HPV-16, also some of the related one, in particular HPV 31, 33, 58, and so on. And within the species alpha 7, besides HPV 18, we do see other members of the family, like HPV 45 and so on. Now, on which parameters are based the oncogenicity? Is not only the identification in cancers, but also the capacity of binding and degrading the P53. In fact, as we can see, so most of the activity is due to is associated with the two groups, the two species, the 7 and the 9. But because the risk has been calculated not only on the detection in cancers, but also on some molecular association, so we know just for few of them very well their oncogenic risk. For most of them, we do not know what is at the moment the risk. So we have a lot of them which show an undetermined risk. The type of infection is that essentially, well, very often the data are projected like this, but so indicating that the cells that must be infected are the basal level, and that you need some kind of scratch in order for the papillomavirus to reach the baseline. Actually, this is an artifact, I mean, meaning that the infected region is always the transitional zone. And the transitional zone between the endocervix and the ectocervix is actually characterized by a monolayer, monostratified. So you do not need necessarily a scratch, because in that region the basal cells are actually exposed. But beside now the infection stage, then we have two types of infections. So the productive one, and in that case, you will see the different expression of the proteins, including the late one for the capsidic proteins, or the abortive one. In that case, the virus is integrated, usually at the level of E1, E2, and in that case, you do not have any further expression of the late proteins, but just of the early one, in particular E6 and E7. Now, the infections we show as a progressive one, but it's not like that. Most of them will actually clear. So you have a main process of regression of the infection. Just a very little percentage will actually progress, and 10 years later, you will see that some of them will start to induce an invasive lesions, which eventually, 30 years later, will induce some kind of cancers. So it's a very minor component that will eventually progress. And so this is a typical historical history of the infection, meaning that most of them are actually regressing, are clearing, and there is even at the precancer stage, like some early lesions, which or dysplasia, will eventually regress, most of them. So the problem is to define the carcinogenicity of this human papillomavirus, and this is what we try to do with Mark Schiffan and Gary Clifford at IARC in 2009, while we were evaluating the infection levels. And it was clear with the data available that besides the oncogenicity that we could determine at that stage, we had another level of oncogenicity, which is related to something else, meaning that during HIV infection, some of the what are considered the low-risk viruses eventually will determine persistent lesions with the potentiality to progress to cancer. Now, this is just to emphasize the fact that HPV-16 and HPV-18 play a major role. Actually, HPV-16 is the one. in not only in the cervical cancer, but also in penile, vulvar, anus, mouth, oropharynx, and this is just a combination of the data of all of them. And this is to show that we have a major difference switching from HIV negative women to HIV positive. So in HIV positive, also some of the low-risk HPVs will determine a persistent infection, and some of them actually we will see in advanced lesions. Similar data has been reported also in other countries, for instance, Nigeria, where you can actually see that in a different population, we have a different distribution and prevalence of genotypes. And in fact, now you can see that in Nigeria, for instance, HPV 18 is the prevalent one. Again, in the positive people, you start to see different types which are not usual in the negative population. So there is a switch in genotypes present in the population. Now let's see some of the data from India. Actually, I was lucky because a few days ago was published some of the HPV-related disease report from Barcelona. And again, you can see what is the first peculiarity. In India, we have is biphasic. So we do not have just a prevalent peak at the lower age and then goes down. Actually, it goes up also around 55, 64. We do not know why. In the Western country, this implies that there is some kind of promiscuity, so that there is some new relationship going on. We are not sure about India. But now something else, the distribution of genotypes in India is very similar to what we see in the Western. So in the normal cytology, we have approximately 5% of mainly HPV-16, and as we go in the more aggressive stages, we will see mainly the HPV-16 followed by HPV-18. A similarity is also for the types distribution in different types of cancer. For instance, in the adenocarcinoma, also in Western country, we have a high prevalence of HPV-18 in comparison to, for instance, squamous cell carcinoma, where the major role is played by HPV-16. We did not have any data on this report on the presence and the distribution of HPV genotypes in HIV women in India. But we do have some other data from West Bengala. This is what I was able to find. And again, what we see is the biphasic distribution. So not only a peak in the early age, but also in the group of 36-40 years. And again, we see the majority of infections in HIV positive. Also in India, in this article, we see that there is a different distribution of types, comparing just the general population. So as you remember from the previous slide, we have mainly HPV-16. In HIV population instead, we have the majority is HPV-18, like in Nigeria. And we have a vast increase in prevalence also in other types, which are not very frequent in the general population. Now let's switch to the cervical cancer. For the cervical cancer in Italy, we have approximately around seven cases among 100,000 people. And if we take the distribution for towns and for regions, then we see that we go from 10 in 100,000 to 4 in 100,000. This is the incidence. The mortality is around 1 in 100,000. Now, when we go to India, well, first of all, cervix cancers is just the second, following breast, and is 11 for 100,000, so it's almost 10 times more. And when we have the distribution by age, then we will see that in the 45-74 years group, we have over 50, the incidence of 50 in 100,000 people. And when we analyze the role or the burden related to premature deaths and disability, then cervical cancer becomes the most relevant cancer in women. Now, in respect to the screening, well, I am not sure that I can pronounce correctly the last name, but Sankaranarayanan, or we call Sanka. Sanka described in 2007 in the Tamil Nadu that just doing once the visual screening for cervical cancer was able to reduce the death and the cancer's frequency in that population. But 2 years later, in 2009, we can see that actually the VIA or the VIA is not actually sufficient. The major role or the major preventive role is by the HPV testing, where we do not have any death from cervical cancer in the negative group. So the problem is how to screen these patients. Well, because it's just a little minority which eventually will progress. And also in the more oncogenic, in the HPV-16, just 1 in 500 infected people will eventually progress to cancer. So at the moment, there are different clinical guidelines, and also in the United States, we have just interim clinical guidance, meaning that they have not decided yet how to proceed. Because before, we had the cytology for the screening and validation by colposcopy. Now we are doing the primary screening by HPV, then we have triage by cytology, and then eventually colposcopy. And this is the flow chart that is now used in Europe and mainly in Italy, following an Italian group from Franco et al. In Lancet 2014. So we have HPV screening, cytology, triage, and colposcopy. And the major problem is for the people who are HPV positive and the cytology is negative, because we really do not know how to deal with them. Because we do see in 3 years intervals that the one HPV 16-18 positive with the normal cytology will eventually progress 40% to CIN3 or advanced lesions. So we are just focusing now on this specific component, HPV positive, cytology negative, what to do with them. This is the study that we are now starting, and will be focused mainly on this group, so positive for HPV and negative for cytology, and we will try to use implement what we have been doing for many years, including the different types of molecular characterization of virus and some of the cellular genes in particular. So we will do the phylogenetic analysis, some of the integration studies on HPV-16, and in particular the identification of the location of the integrated regions, and some of the study on genes which have been shown to play a role in developing cancer, in particular mutation of the TP53, and more recently mutation in a non-coding region of TERT promoter, which has been identified in over 20% of cervical cancer, penile cancer, and above 6% of head and neck.
Dr. Buonaguro, can we wrap up? We are running late.
Okay. I think that probably you should discuss if it's relevant to, or there is any priority in doing some of the screening in a more sophisticated one, also in the Indian population, given the high incidence of cervical cancer, and eventually how to proceed. Probably the DNA molecular characterization would be, for papillomavirus would be the best choice.
I wanted to highlight that we've done studies characterizing the virus at National AIDS Research Institute among HIV-infected women. And we have found, as you have shown, that there are many other viruses other than 18 and 16 that we've identified, not only in the cervical region, but also in the anal region, 51 and 53, and some more. So basically, I wanted to check with you, if we have to go about looking at the oncogenic potential of these viruses, what would you suggest? That was one. My second quick comment was about how, when you pointed out that women are dying earlier of cervical cancer in India, a recent registry match study that we did showed that women with HIV had a 15.7 population-based study, showed that they had a 15 times more incidence of cervical cancer as compared to other women. And that probably shows why younger women showed an increase in mortality in the more recent years. Thank you.
Well, I think that India is probably in, for this specific subject, would be probably the country where to study this phenomenon. Because in other developing countries, what we call developing countries, I mean, I would not call developing countries India, but anyway, in the other countries like Uganda, Africa, and so on, they do not have the mean or the technology to analyze this type of aspects. And in the Western country, we do not have so many people where to do the study. So probably I can offer to collaborate, or you can do, I mean, you have the technology to do it in any case. And probably we have, in India, we have the opportunity to restage, to reevaluate the low risk in the healthy people, because in other conditions, particularly on HIV, probably the risk is totally different, and we have to analyze how to. We should study how to characterize this type of risk, how to calculate the risk, because we do not know, and we cannot do in any other place, because the other countries, they do not have the technology, and or they do not have the numbers to do this type of study.
Thank you.
Thank you, Dr. Franco.
Hello, can we have the
yeah.
questions at the end of session?
Yes, please.